Alright, we learned from the previous video about water potential, and now I like to remind you again about what is the water potential, especially in a plant cell. Water potential is the tendency of water to move, and it has two components in the plant cell: solute concentration or psi S, or solute potential or osmotic potential, and pressure potential or psi P, and in our case is the turgor pressure. Psi W, but W is equal to psi S plus psi P. Solute potential is the potential of water to move from one to another cells as influenced by solute concentration. Again, if you have a lot of solute, then you have negative values of psi S. Pressure potential is a turgor pressure that develops against the wall as a result of water entering the vacuole of the cell, or just think about water entering, forget about vacuole. And the higher the pressure, the more positive psi P. All right. So let's take a look at how the plant cells is influenced by osmotic condition. Here we have three conditions: under the osmotic pressure, under equilibrium, or under water loss condition. These cells has lower water potential compared to the outside of the cells. What does mean? It has a lot of solute, then water from outside move to inside. And if you notice carefully, this rectangle is vacuole, one of the organelles inside the cells. The main function of it is to store water. So under the osmotic pressure, water move in, and the cell is under hypertonic condition, or we can say the cell is turgid. At equilibrium, water move in and out is equal. You can say that there is no net of water movement in and out. So this cell is very happy under normal condition. The cell is called isotonic, or the cell is flaccid cells, if you like. Finally, you have water loss condition. At this condition, the water can move out of the cells. Why? Maybe because outside you have more solute, for example, more sugar, more salt, more amino acid, for example. Then water inside the cell has more concentration, then it moves out. Or outside you have a lot of positive pressure push against the cells, so the water move out. We can call this condition that the cell is under hypotonic condition. Hypo means less, hyper means high. And the cell is considered to be plasmolytes. And the process to make this plasmolyte cell is called plasmolysis. If you may recall, in the lab one or in the lecture one of plant cells, we made protoplast from the plant cell by applying salt to outside of the cells. Then the cell shrink, and you can separate the protoplast from the cell wall. This is the same thing. And this table, I summarize the direction of water movement in under turgid, flaccid, and plasmolysis condition, and also water potential inside compared to outside of the cells. And this is an example of flaccid cells and plasmolyzed cells in the real plant cells. All right, I like to stop right here and we will continue with the next video on imbibition and active transport.